So I'm gonna make a series of me probably reading a lot of research papers. And of course, I'm just gonna make a series. I'm gonna read with us all together, and just everyone so that everyone has an idea of what I'm researching about. Yeah. So this is a how information theory handles cell signaling and uncertainty, published in the year 2013, which is a long time ago on PubMed, a very famous journal published place. Anyway, so abstract. Information theory allows analysis of cell signaling capabilities without necessarily require detailed knowledge of the signaling networks. Um, not well. The thing is, what I was thinking is that right in the beginning, it say you don't really need require the detailed knowledge of a signaling networks. True and not really true, because if you really don't know the networks, you don't really cannot. It's hard to decode the information. That's what I'm thinking of, and my research is more about like, hey, I do really need to know the networks, and I do really need to know what the information is about, because you really want them to you want to deep down like know as much as you can. But anyway, let's see what this article is talking about. Intracellular biochemical network have traditionally been studied by simulating population of genetically identical cells and measuring the aggregate response. However, such population-based measurements may obscure the idiosyncrasies of individual cells and therefore suggest deceptively precise input-output relationships. Consequently, signaling pathways have been viewed as the finely tuned circuitry that programs the cell to behave in a predefined manner. Detailed study of cellular biochemistry at the single cell level now show that cells responding in masse may have quite varied behaviors when examined individually, raising the question of how precise signaling pathway can control cell actions. That's interesting. Um, that's interesting. It is likely that under most circumstances, cells in populations constantly diversify their states. Thus, for example, the response of their signaling networks. Thus, there is an uncertainty in signaling outcome, and responses of cells randomly selected in the population are unpredictable. In this sense, the match between the environmental input and the cellular output can no longer be predefined and stereotypically precise. This hampers the ability of the individual cells and small cell enabled to make appropriate decision in fluctuating environment and require a fundamentally different view toward analyzing signaling system. Hence, rather than relying on similarly robust and sensitive signal input-output dependency to analyze network and cellular behavior, we should instead seek to learn the limit on how cell signaling can enable decision making, given the cell uncertainty response to changes in the environment. That's just interesting. I mean, this the last like sentence that here, the last sentence here is really what I've been trying to argue for a whole time. Um, we should seek to learn the limit on how cell signaling can enable decision making. Now, really, like if you're able to send, like, let's say you have an imaginary signal and you trying to send into the uh um cognitive, right, structures, and you want to study the cognitive structures, right? Really, like, you really want to know the limit, because this will really try to define what cognitive really mean, what cognition really mean. I mean, right? We're really defining that. If you're able to do the input and output scenario, right? Signal and input and output dependencies or scenario thing. That would just mean, do I rank this thing in a hierarchy? I mean a higher like form, or is it lower form? It actually depends. To be honest, I would say like this is really my approach to actually learn about cognitive structures. We want to find what is this, right? I want to rank the cognitive structures and therefore trace out the um what is the lowest form of cognitive structures. Which is this is exactly what I've been trying to tell all the time too. And also like it does mention about between the environmental input and cellular output, pendular predefined and stereotypical precise. This is exactly what I've been trying to tell to people as well, that sometimes environmental impact is true, but sometimes you may not necessarily need it. Yeah, variability in cells response is frequently referred as a noise, and the current metric to characterize noise report on its magnitude but do not quantify how the noise limits a cell's decision-making abilities. Indeed, performance of a cell signaling network depends on more, which one? Then more than just the level of noise in its underlying chemistry. For instance, signaling may allow a population of cells to simultaneously sample several distinct classes of behavior, a type of cellular bed hatching which can improve some aspect of decision making, but with a cost of increased variability. Therefore, a new language may be needed to understand and quantify the impact of noise on the cellular functionality. That's exactly what I'm trying to do as well. Um, well, wow. Okay, I have been thinking about this on my own. I did not read this paper at all beforehand. I've been thinking along by myself, and I just can't believe that there's someone just really try to underline this. Uh, and the performance of a signaling network depending more more than just the level of noise in its underlying chemistry. For instance, signaling may allow a population of cells to simultaneously sample several distinct classes of behavior. Okay, so the question I'm having that I'm struggle with is really what would be how would you right how would you do the math and how would you modeling this? That's the that's the wall I had to break. But let me just continue. Oh, mathematics. Mathematics turns out to have just the right theory. This theory has already been adopted to understand the workings of another type of noisy signaling network, the nervous system. Created to analyze uncertainty in human communication, information theory enabled the limit of decision-making fidelity to be rigorously defined and measured. Conveniently, its general formulation permits analysis of many complex systems, including those found in biological signaling. Within this theory and in the context of signaling, information is quantified as the uncertainty about the environment that is removed by signaling activity, which is equivalent to the knowledge gained by the signaling system. The amount of information depending on both the amount of variability in the environment, the initial level of certainty, and the noise in the sampling process itself, affecting the amount of uncertainty remaining. Extending this definition, we can also determine the information capacity. Oh my god, I can't read. Of a system, which is the maximum information that a signaling system can obtain about some aspect of the environment under ideal condition. This capacity is intrinsic property of a signaling system as much as underlying chemistry that is key determinant of the achievable decision-making fidelity. Information making fidelity rigorous defined. Um, I mean, true, but still, it doesn't really quantify the problem. Doesn't really solve because when you want to modeling a signal, you really wanted to display the information as well, right? Rather than um uh rather than the quantitative, right, variable or or the variable and all the things. You really want it to display the information, right? That's the challenge in the mathematics. Like, how do you quantize, or how would you quantify the amount of information? Or I mean, if it's not about quantifying that, how would you really displaying the information uh from the from the given energy, or give in this case a given noise? I mean, okay, let's just try to continue. As an example, consider a signaling pathway whose output measure the concentration of an extra cellular ligand. I mean, those response. Signaling noise prevents a cell from determining the precise ligand concentration. However, does the noise also prevent a cell from resolving different concentrations of the ligand? And if so, how many and how accurately? Information theory state that it is possible to use noisy signaling output to accurately discriminate different input doses. Interesting. Interesting. I mean, this is interesting. I didn't know this. I will have to look up this. There's a reference here. Huh, interesting. Furthermore, the number of resolvable concentration is limited and is symbol function of the pathway capacity. Alternatively, in mistake to occur, the capacity determines the minimum amount of error that a cell must tolerate, with high capacity and ambiguously allowing for the lower error. Information theory allows such category statement without necessarily requiring detailed specifics of the signaling network organization and operation, and thus can be used to analyze the capacity of complex and incompletely characterized biological systems. I mean, okay. Currently, we do not understand the decision-making limits of the vast majority of signaling system, even those affected by variability. True, true, true. That's what I've been talking about as well. Consequently, the factors that affect and regulate those limits are also generally unknown. That's true as well. Thus, from the standpoint of information transfer, it is essential to determine the capacities of this signaling pathway and network and the relationship between the system structures and capacity. For instance, information loss at each step is processing through should prevent information sources and destination from being separated by more than a few intermediates. Okay, sure. Simultaneously, it is often necessary to integrate multiple pieces of information within the cells. Integrate multiple pieces of information within the cells. Both of these considerations drive the formation of so-called smaller networks that widespread in biological systems and other networks in which a relatively short path connects any two signaling nodes. Such networks are configured so that the multiple signal pass through central nodes, thereby raising the information theoretic question of how the signal are multiplexed through the hub so as to minimally interfere with one another. I mean, uh, yeah, the question is for the math, like, is it an equation or is it an algorithm? Right? That's what I've been debating because there's a lot of things that you can't really equate them. I mean, like, you cannot like put like F is equal to ma, just say, when I was trying to develop the math for that. It's rather than um do you want it to be an algorithm or you want it to be an equation? I don't think an equation works. I would try that, and I don't think it worked out really well. Um yeah, so the thing is, if an algorithm, how would that gonna be look like then? It's not that easy at all. Acquiring information typically costs the cell energy, time, and opportunity, so the signaling system can collect more information that is necessary or ignore information that is easily obtained, wasted valuable resources. Therefore, under evolutionary pressure, it is expected that signaling systems are optimally matched to the sources of the information they have evolved to process. Indeed, examples from neuroscience, such as sensory perception and developmental biology, such as embryonic patterning, show that biological systems usually have the capability that is minimally sufficient for information they process. Huh. Okay. So what information? What was that? Okay. I want to know. I want to know now. This optimally principle can answer long-standing question that cannot currently be addressed through model or direct experiments. One example is determining which of several putative aspects of environmental input, ligand dose, rate of dose change, or duration of ligand presentation, are biological relevant. Information processing ultimately suggests that the aspect of the input associated with the higher capacity is more than pertinent one. This concept further implies that the condition that maximize utilize the information capacity of a sensory system should reflect the natural fluctuation in the environment. This condition can be computed from the control laboratory of observations, enabling to form an inverse ecology that is sometimes the only visible way to gain insight into the cell's natural surroundings. Similarly, argument can be used to infer which aspects of response to environmental input is most relevant than to the output. Um, I see what it's trying to do. I see what it's trying to do. So your input is gonna be your so the thing is, I think is what they what like I think this is what based on what I'm trying to understand here is that the input is really the information. The thing is, the signaling that they are doing is really relying on the um, environmental condition for the cell to send signals. Now, my research is more about you don't rely on that. Is there a way you can trigger the cell to signal within themselves? Is there a signal that triggers the cell to signaling within themselves? That's what I'm trying to really researching on. They are on a different route. I think I see what they're trying to do. Then complete different route. They use the environmental input to stimulating cell behavior, which is a way, but does it help though? Like, okay, so experimental laboratory, so you put in the cell within that environmental dish, and then you see the cell response, and then you map it out. Uh, to determine the frequency in which a stimulus, such as ligand, occur in the cell's natural environment, a population of cells genetically identical can be exposed to a ligand in a experimental setting. Individual cells' responses can be measured at a range of doses. That information can be used to discover information about cell natural environment. There is variation response at which dose or noise. Mathematically, such experiment measurement have provide a condition probability distribution P of response to a signal. It is assumed that the cell is optimized to obtain the most information about frequency of doses it encounters its natural environment. P of next signal. Environmental information theory, IT, determine the P signal that maximizes the information capacity of a signaling network that is responsive to the particular ligand. This reflects the dose frequency in the cellular environment, a natural environment. Um, sure. So you they want to see the this one. They really want to see the response so that they map it out to the information from the environment, which is one way. I didn't think about this, which is smart. I didn't think about it. Could be done though. As biologists are finding more uses of information theory is evolving to handle more complex biological network by rigorously quantifying the probability and limit of a cellular information transfer. New question will be formulated and answered within the cellular paradigm shift that is already underway. As the ability to quantify the functionality of a signaling network improve, we will hopefully gain insight into detail of the underlying chemistry while gaining a deeper understanding of high-level organization and functionality. With very good article, but the thing I really want to know is that this model right here, right? What response are you trying to measure? What is the response in here that you're trying to measure as well? Is not really clear. I have to say, is there anywhere mentioned in here? I guess. Do they say in response? To create multiple information of cells, consideration acquiring knowledge to be cause the cell up to signals. Ignoring both example. Not really, right? I don't really see any response on mapping this outcome here. And you're in this one here, the only way you can study or learn is the the environmental surrounding of the cells and then the signal of the cells. That's really good. The only problem here is that the only limit side that I'm seeing is that you don't really know what the signal is about. Because if you really can narrow down what the signal is really about, then you can actually learn a little bit more about evolution and cognitive structure as well. But I don't think this is the main focus of this paper at all. So it is what it is. But it's a very good paper too. Something I know how to note it to myself. Yeah, pretty good paper.